In the first lecture, we have talked about interphase, that is G1, S and G2. Today, we will discuss mitosis in this second lecture. Now, mitosis. What is mitosis? It is the type of division where the chromosomes will replicate and get equally distributed between the two daughter cells. It is also known as equational division, and it's mainly responsible for growth and repair of the cells. Equational division, as I already mentioned. Why equational division? Because the number of chromosomes is the same in parent and daughter cells. Now, the process of mitosis can be divided into two parts broadly, that is karyokinesis and cytokinesis. In karyokinesis, what will happen? The nucleus will divide first. Only the nucleus will divide of the cell, and then it will be followed by the division of cytoplasm. Each part of the cytoplasm will enclose one nucleus out of the two. One nucleus out of the two. As the nucleus divides first, then after that it will be enclosed within the cytoplasm. Now, here you can see the cell cycle. So here the cell, okay, this is the cell cytokinesis, okay, it formed two new cells. After the formation of the two new cells, what happens? It undergoes that interphase G1, okay, then S phase and then G2. In S phase, what happened? The DNA replicated. In G2, there is formation of the proteins, growth of the cell, and then it enters the mitosis. In mitosis, what will happen first? It will be karyokinesis. The nucleus will divide into two, followed by division of the cytoplasm. So this part we are going to talk about today. The rest of this part we had already discussed in the previous lecture. Now, mitosis again. So this is the cell cycle. Okay, this is the cell cycle. The cell it undergoes interphase. Let us say from here, it undergoes interphase, it undergoes, and then it starts with mitosis, the cell division, the actual cell division. This is the cell, okay, which is ready for dividing. It will undergo 4 phases. We have divided for our convenience into 4 major phases: Prophase, Metaphase, Anaphase, and Telophase. At the end of Telophase, we get the karyokinesis is complete. 2 nuclei are formed. These 2 nuclei will now, what will happen? The cytoplasm will divide here, which we call as cytokinesis, giving us 2 new cells with individually one nucleus. So then each of these cells will again undergo the interphase, and again it is ready for passing through prophase, metaphase, anaphase, telophase, again give 2 new cells. So this way the cell cycle continues. So now we have to learn details about each of these parts. In first interphase, okay, in interphase, the cell, what is happening? Everything is growing. This is the centriole. This is an animal cell we are talking about. This is the centriole. These are the chromatids. This is the nucleus and the cytoplasm. The cell membrane, other cell organelles are present here, though not shown. After interphase is complete, it has moved through G1, S and G2. It has double the DNA content, because during S phase the DNA has replicated. Even the cell organelles have increased in number. Now what will happen? First, this, which was a long thread-like structure, this chromatin, it will arrange itself into its individual chromosomes. It will arrange itself into its individual chromosomes, and these chromosomes will now be distinct. They will be broad and distinct. Now, after this, which we call as the prophase. In the meantime, the centriole, it will give rise to its 2 centrosomes. It is made up of 2 centrosomes, as we already know, 2 centrosomes. Now, what will happen? These chromosomes, okay, these chromosomes will get arranged into its 2 chromatids. If you can recall, when we were discussing about the cell, we had learned that each chromosome is made up of 2 chromatids. These 2 chromatids which are joined together with the centromere. These 2 chromatids are what? These 2 chromatids are those replicated DNA. Okay, these are the same. These are just copies, these two. So they will be, the chromatids will appear in the prophase stage. After which they will move towards the central plate, okay, central, and in the meantime, of course, the nuclear membrane has disappeared, and these chromosomes, they are simply lying in the cytoplasm. Now what will happen, these chromatids, they will line themselves in the central space of the cell. Then here the centrioles, they will give rise to some fiber-like structures. They will give rise to some fiber-like structures which are known as spindle fibers. Now the spindle fibers will pull away the chromatids of each pair. Each pair, the chromatids will separate out, move towards the two sides, and then there will be reformation of the nuclear membrane. That is interphase, prophase, metaphase, anaphase and telophase. Metaphase, arrangement in the central plate. Anaphase, pulling away of the chromatids. Telophase, aggregation of the chromatids. Now, furthermore, prophase. What happens in prophase? Chromosomes condense and become visible. Spindle fibers emerge from the centrosomes. Nuclear envelope breaks down. Centrosomes move towards the opposite side. So the centrosomes, they will move towards the opposite sides in the prophase. Prophase. Prophase, again, this is the most lengthy part of the cell division. So for a convenience, sometimes prophase is also divided into two parts, early prophase and late prophase. In early prophase, there is condensation of the chromosomal material. Then the formation of the spindle fibers from the centrosomes, and towards the late prophase, the nucleolus, the nuclear membrane, everything starts disintegrating or breakdown. Even the cell organelles, they start disappearing at this stage. It is followed by metaphase. In metaphase, what will happen? That each chromosome which is made up of two chromatids, they will remain here. Now what will happen is, these two chromatids, they will, each pair of chromatid, they will align at the central space. This is the central space. They will align themselves here, align themselves in the central space here, in any direction, but centrally usually. And the centrosomes, they have given out the spindle fibers. These are protein fiber-like structures. And in case of plant cells where centrosomes are absent, it's directly from the cytoplasm, such spindle fibers will appear. And these spindle fibers, one end, it is joined with this point, centrosome, other point is joined with the centromere of the chromosomes. So chromosomes continue to condense in metaphase. Kinetochores appear in the centromeres. Kinetochores are disc-shaped structures near the centromeres. Mitotic spindle microtubules attached to the kinetochores. These spindles, they attach to the kinetochores. Chromosomes line up at the metaphase plate. Here they line up. Each sister chromatid attached to spindle fiber originating from the opposite poles. So here these two are known as sister chromatids. So what will happen? They will be attached to the spindle fiber here. They are attached to the spindle fiber. Then in anaphase, next phase is anaphase. In the meantime, what is now, of course, you have to always remember there is no clear-cut demarcation of one phase to another. Just with the major events, whatever is happening, we mark the phases. The next, what will happen, as the spindle fibers have attached to the kinetochores, then this will shorten. As they shorten, there is separation of each pair of chromatids. They separate out. Centromeres split into two. Sister chromatids, now they will be called as chromosomes, then move towards the opposite poles. They move towards certain spindle fibers begin to elongate the cell. So now what will happen? They will move towards the opposite poles. Then in telophase, what will happen? In telophase, now the chromosomes arrive at the opposite poles and begin to decondense. They begin to decondense now. What will happen? They will again, whatever condensation has taken place, again they will start to dissolve and become one single fiber. Then nuclear envelope material surrounds each set of chromosome. Each set of chromosome is covered by nuclear envelope. The mitotic spindle breaks down. The spindle fiber will no more exist. Spindle fibers continue to push the poles apart. Now these two will move further towards the poles. So it completes your karyokinesis, prophase, metaphase, anaphase and telophase. So what happens here? We have got equal number of chromosomes on both the sides, which started with, like, say, one chromosome which became two chromatids. So one chromatid moved here, other chromatid moved to the other pole. So ultimately what will happen? Each will be having one. Okay, so that way the chromosome number is maintained. So then after that it is followed by the cytokinesis, which will lead to formation of the two new cells. Then furthermore, next we will move towards lecture 3. That is, it will be our next lecture where we will discuss about meiosis. Thank you.